Hi, my name is Rachel Larcher. Thank you for coming back to the HPE Part 3. This one will focus on new research done by it, done for holoprosencephaly. So, currently for treatment, there is no general or comprehensive all the way around treatment. It's more specific to each patient. Commonly, a whole team of experts is assigned to give treatment to one patient. They'll be experts in medical specialists, medical surgeons, plastic surgeons, developmental specialists, whatever is needed for that individual person. And these don't actually directly target HPE. It's just more used for easing some of those symptoms. There are current studies, however, that are being done to try and see if they can at least target a specific kind of HPE. As we learned before, there are different kinds, and a lot of them are caused by a lot of different genetic factors from chromosomes all the way down to just single genes. There's a study right now that's going on currently. It's called the retrospective study using high throughput sequencing, HTS, on biological samples to improve genetic counseling for patients with previously explored craniofacial midline defects. It's a long name, but essentially what's happening here is they're taking pre-existing samples and using next generation sequencing to try and figure out and target a couple of different genes, especially with gene editing tools. This started in February of 2021, but they're still developing results. No results have been released yet, but it's the most current study I could find on HPE for its causes and what they can do for treatments for it. Now, next generation sequencing, which is being used in the study, also called NGS, is a DNA sequencing technology. It uses revolutionized genomic research to sequence the entire human genome. Now, this can be done with NGS in a single 24-hour day, whereas the Sanger sequencing, which is the older kind, can take up to a decade to get the full human genome back. However, the NGS is not used routinely because there's a lot of technology through it, and so it's not used in clinical trials. It's just more for studies. There are several platforms that can use it, though, which is really cool. The NGS process, it is taking millions of genetic information and then sequencing it in DNA fragments in parallel. So it'll take millions of genetic information and sequence it at the exact same time. And it'll sequence it into small fragments. And it does this, each base pair is multiplied several times to provide more high accuracy in the results. And to allow for looking for those genetic mutations. These fragments are then pieced together through bioinformatics analysis, and then this process can be used for the whole human genome or just specific regions or bases or sequences. The main flaw in this system is, like I said, the bulkiness of the equipment. It's just too bulky. It can't be put in every single clinic, and so it's mainly just used for research purposes. Now, can we cure HPE? As of right now, the answer is no, but science is constantly upgrading, and so I'm sure that there is a way that we just don't know of yet. HPE occurs while the child is still in the womb. So what it is the brain fails to develop properly inside the womb. So it's nothing that can be fixed after the child is born. When the child is born with a defect, it's just how it is. The hindbrain does not develop into the forebrain, which means the prosencephalon does not form. There's no separation between the right and left hemispheres or between the cerebrum and the cerebellum. This causes developmental delays and craniofacial abnormalities. So, how do we cure it? If it causes all of these things, we're not exactly sure what causes it. for each person, and there's a million factors that can cause it. Well, one thing that we have been looking into recently is CRISPR technology. CRISPR stands for clustered regularly interspaced short palindromic repeats. These are over thousands of years old, but they were first discovered in 1987 in the E. coli genome. So they've been existing for over thousands of years in E. coli and other DNA in other species, but they were not discovered until 1987, and they actually were not even used until 2008 for things such as gene editing. They're small repeated sections of DNA, and it's found that the palindromic repeats could be altered and that DNA would code for the fixed gene. So when the cells divide, the RNA would translate the edited gene. The CRISPR method consists of taking a particular gene and cutting it with the Cas9 enzyme. It's then fixed by substitution of a base or bases through a repaired template strand. So when that whole thing with the repaired template strand is inserted back into the cell, the DNA strand will bond and code for the new strand, the new template strand that was created. So how do we use CRISPR to treat HPE? Well, it would have to be done in utero. So it would have to be done while the child is still in the womb. If there were any way that we could detect it early on and know when the brain is not developing properly, if we could see if that was truly HPE, then we might be able to use gene editing if we could find the exact gene or the exact chromosome that causes it. A lot of times, trisomy 13 is what causes HPE. Well, gene editing can be done to help get rid of that extra chromosome, or just fix those couple of genes here and there that could cause HPE if we knew exactly which one it was in each patient. Then we could test some of the amniotic fluid, we could test some of the stem cells in the child or some of those cells, and then we could use that and use the DNA from those cells of the zygote to create a new template strand using CRISPR technology and those short palindromic repeats. And then when we insert that fixed strand back in, it might be able to help it. Now, there's no studies going on about this right now that's says that this could be a thing, but this is just what I'm thinking could possibly happen if we get more developed with CRISPR and find out a little bit more about HPE. But as far as that goes, that's all I have for you. Thank you for listening. If you've listened to all 3 podcasts, thank you so much. And I really do appreciate any feedback that I've been given so far. This class was really fun, and I'm super glad that I was able to take it with the people that I had, and I hope to take it other classes with them in the future. Thank you for listening.